Okay. Well, welcome everybody. I'm glad you could join us tonight. Hope you're not getting too many storms wherever you are. We're under a severe thunderstorm warning here in Monroe in central Union County. But tonight you're in for a treat. We have Linda Chalker Scott. She has a PhD in plant physiology from Oregon State University, and she is the Extension Urban Horticulture Specialist and Professor in the Department of Horticulture at Washington State University. She is the creator and manager of the Garden Professor's Facebook page and the Garden Professor's blog. Her website, The Informed Gardener, is a repository of science-based white papers, case studies, podcasts, and other curated materials. Early in her career with Washington State University, Linda served as the Master Gardener Program Curriculum Director, and she was responsible for the development, revision, and delivery of curricula to the Washington State University Master Gardeners. She's well known and widely respected for her work. Welcome, Linda. We're so glad to have you tonight.
Thanks, Debbie. I really appreciate being here. And I'll apologize to everybody for the quality of my voice. I was at the meeting in Iowa 3 weeks ago and brought home not only good memories, but a bug. And so I've been getting over the bug part, and I feel fine. I just sound really bad. So you'll apologize for my hoarseness. And I do understand that all of you have a handout for the talk, which will help a lot in case you miss something or want some little extra information. There's some good information on that handout. Okay, so I've been doing horticulture myth busting for almost 25 years now. And I'm going to preface this by saying that as I was getting my PhD in the late 1980s, many of the things that I'm going to be busting tonight are things that I was taught and things I actively did. So if you see some of your favorite practices or products skewered tonight, be aware that we're all in the same boat. We're all learning new stuff. But the really cool thing about both urban horticulture and arboriculture is that the science is young, it's quickly evolving, it's dynamic, and it's a lot of fun to keep up with. Okay, so anyway, as this picture shows you, I I try to attack various myths and shoot them down, and like zombies, they just come back, sometimes even more virulent than ever. So I really rely on master gardeners all over the country to help spread the good word and make sure that we're teaching good information. So what I'm going to start with today is just briefly going over how to evaluate information. And specifically, we're going to be looking at assessing products and practices that are associated with gardening and landscapes, as well as phenomena. And this one is maybe kind of a odd one for you. You'll see what I mean when we get there, and it really is, it's pretty fun. So I'll spend a little bit of time doing that, and then we're going to jump into how you can look at peer-reviewed papers. And peer review really is the gold standard, but sometimes you're going to find stuff where peer review doesn't work for some reason. And so we have to be aware of that too. So we can't just assume that just because something gets published, it's necessarily true. And finally, we'll have time at the end for questions, and I'm happy to stay on as long as you all have questions and as long as my voice holds out. So the first thing I want to make you aware of is a publication that a colleague and I did for Washington State University a few years ago. And this is, it's a free peer-reviewed downloadable manual, and it helps non-scientists learn how to evaluate information really on any topic at all. It explains what the scientific method is, how you put an experiment together, and then, just some ways to test information and some comparative stuff. There's a whole bunch of interactive things on the web. And it really is a nice publication to use not only for master gardeners, but for them to share with their clients. So the first thing we'll talk about is what I initially called the credibility objectivity sieve, but very quickly it morphed into the CRAP test, which is much easier to remember. And what it basically boils down to is you take your raw material, your data, your information, and you put it through the sieve, and maybe you'll get gold or maybe you'll get crap. And what CRAP stands for is credibility. So in other words, how credible is the source? If it's an author Are they experts in their field? If it's a publishing house, do they use peer review, or is it just anybody can write anything? Secondly is R for relevance, and relevance means, is the information relevant to your activities as a gardener or landscaper, or is it really something that's geared towards agricultural production? And they are not the same thing. And so we can't just take things that are used in crop production and assume they're going to work for managing gardens and landscapes. Third part, the A is for accuracy. And this basically means, is the information the most current, reliable information that's out there? And there's a lot of older papers and work that are still relevant, and a lot of those are seminal papers and important discoveries all through science. But as I said earlier, both urban horticulture and arboriculture are very quickly evolving. And what you learned 20 or even 10 years ago isn't necessarily what's true now. So we need to really keep track of the current research and make sure that what we're using for ourselves and for our clients is up to date. And finally, purpose. And so what I mean by purpose is, what is the purpose of the speaker or the information that you're reading? What is the author's purpose for sharing that? Is it for disseminating information? You all are lucky because you work with Debbie and other people in an extension, and our mission is education. So you know that
Uh-oh.
I got muted apparently.
I did that. I am so sorry.
Okay, can you tell me where I cut off?
Like about a minute ago, less than a minute, 30 seconds.
Okay, so hey, I started talking about purpose.
Yeah.
Okay, I'll just start with that again. So the purpose of the information is the author or the source trying to share information and educate you, or is it trying to sell a product, or is it trying to push an ideology? You know, what's the purpose of the educator or the material that's being used? So when you're answering those types of questions, you can figure out whether this is really good, valid information for you as a master gardener, or whether it's something that you can just easily set aside. So what I've done over the years is kind of categorize products and practices and phenomena into categories such as no supporting science. And so this means, and unfortunately, this is the largest category of all, but unfortunately what it means is that this is where you have either no research, and you'd be surprised at the number of things get passed around as information where there is absolutely no research to support. Or there might be research, but it's inconsistent, meaning that sometimes it works and sometimes it doesn't. Or maybe all the research is negative results. Or maybe it's poor quality research or reporting. We'll talk about that a little bit when we get into the peer review stuff. And all that means is, yeah, there's some research out there, but it's really not well designed, or it's biased, or it's not analyzed properly. Then there's misapplied science. And so, as I alluded to before, agricultural crop production is not the same thing as planting a garden or a permanent landscape. And so products and practices that are geared towards maximizing production of crops are sometimes erroneously used in garden and landscape management. And so we have to be very careful of that as well. And then there's the over-extrapolated science. And so that just means that it's a product or a practice that works really well under controlled conditions. So in a laboratory, or perhaps in a greenhouse, or someplace else where everything is controlled except for whatever the product or treatment is. And it's really easy to show that a lot of things work under these conditions, but when you take them to a landscape or a field situation where you have lots and lots of environmental variables, all of a sudden they don't work anymore. And so we have to look at those and make sure that the things that we are promoting are things that actually been field tested and verified to be of practical application and not just something that's theoretical. Okay, so as I said, the first category, no consistent, reliable supporting science. And this is an abbreviated list, but it includes things like balanced fertilizers, compost tea, soil conditioners, kelp products, the superiority of organic products, vitamin B1 transparent fertilizers, and wound dressings for trees. And so I know you've probably heard of most of these, if not all of these before, but they're things that have been heavily marketed in the absence of any good reproducible scientific data. Then there's practices such as cautioning about not watering when it's hot, biodynamics, companion planting, injection of fertilizers into the soil or sometimes even into the plants themselves, hugelkultur, lasagna mulching, leaving root balls intact when planting trees and shrubs, the superiority of native plants, and permaculture. And so that list I know is something that I'm sure almost everything on there is something as you've heard of before. And one thing you'll notice about it is that, and we'll talk about this, is a lot of the things that you see are appealing at an emotional level. And we have to keep that in mind. A lot of things that where the marketing is the purpose of sharing information, and not the education, but you know, the marketing is the purpose of the information being shared, and we have to be careful of that so that we're not falling into a trap of recommending things that aren't based on science but are heavily marketed. Excuse me. So I'm going to start with lasagna mulching because it is highly popular everywhere in the country, and it's because it's lasagna, right? And everybody loves lasagna. It's a recipe. It's do it yourself, and so we go in with that mindset, that emotional hold, and then we apply it to our soils. So this is something that, as I said, has been going on with greater and greater frequency, and we need to think about what it is and what its actual effect is on the soil. So the good thing I will say about it is that the reason that it's promoted in terms of what it will do for the soil is it prevents soil from being tilled. And we know that no-till is really the way to go with planting gardens and landscapes. Even agricultural production is doing no-till now because of the damage that tilling does to soil and the fact that it releases a lot of carbon into the atmosphere. So no-till is really a smart way to prepare soil, but we need to differentiate between good ways of doing no-till and bad ways of doing no-till. So as I said, it's a recipe, it's a do-it-yourself, it's noodles and sauce, and cardboard are the noodles, and sauce is compost. And so you're told that you can just make this as deep as you want, and then you can either plant into it, or you can just use it to prepare the soil and then move it aside. But most people just let it stay in place. It's very emotionally appealing for a lot of different reasons, as I mentioned before, including things like recycling materials. But the problem is that there is no science, not any science at all. And so we have to kind of take this practice apart piece by piece and look at the validity of using these materials. So corrugated cardboard, as I mentioned, are the noodles for this process. And it's a sheet mulch. Like anything else, it's a two-dimensional mulch, and I'll be discussing those in just a minute. It's going to interfere with the ability of soil to exchange carbon dioxide and oxygen with the atmosphere, and it also interferes with water moving into and out of the soil. And if we take a step back and think about it, corrugated cardboard is used in pizza boxes, and it's used by Amazon. And neither pizza companies nor Amazon would use materials that quickly break down. They can't afford to. So when corrugated cardboard is made, there are compounds that are put into the slurry that will resist water. And so sometimes it's waxes or other types of hydrophobic materials. And this is done to make the cardboard resistant to breakdown. Obviously, it will break down, but it doesn't break down quickly. And the less oxygen there is, in other words, the deeper the cardboard is buried, the slower it breaks down. So it is going to be something that's going to interfere with the ability of soil organisms to get water and oxygen. The second thing to do is think about the sauce, and especially because a lot of these online recipes say you can make this as deep as you want. And the problem is that when you put down lots and lots of compost, what you're doing is you are over fertilizing the landscape, because compost is just nutrients. They're organic source as opposed to an inorganic source. But the soil doesn't differentiate between something that's organic and inorganic. And the more you put down, the higher your nutrient toxicities can become. And something we need to think about in terms of organic matter and sauce is that, when I first learned about the soil pie chart back in the 80s, it didn't look like this. It had the solids as being half of the soil, and it had the pore space, the air and water being most of the other half, but then it had a wedge in there that said organic material. And there was this perception that you had to have between 5 and 10% organic material for ideal organic levels. And that's just not true anymore. The more work that soil scientists have done, the more they have found that ideal organic really is dependent on the soil itself and the environment that it's in. And it can vary heavily from really low levels of organic to pretty high levels of organic, and that's natural for those systems. But there is no simple recipe for how much organic matter is ideal. So because there hadn't been any information at all in terms of research on cardboard or lasagna mulching or anything having to do with that process, we started looking at it from the ground up, literally, by looking at different mulch materials and seeing how they would interfere with oxygen movement and carbon dioxide movement. So this is a very simple experiment, but that's because, as I mentioned before, when you're doing lab experiments, they're going to be simplified so that you have one variable, in this case mulches, and everything else is the same. And so the soil is the same, the buckets are the same, everything's the same. So all the buckets were filled with soil. Some of them were left bare as being controls, and then some of them were covered with different types of mulch materials, as you can see here. And then each of these buckets, and these are, we call them mesocosms, so they're bigger than microcosms, and each one of them has 2 sampling ports, and one of them is above the level of the soil and or the mulch, and the other one is below, and you'll see that in a different picture, but if you see where my arrow is there, you can see that there's a port in that one that's actually below the soil. So you're measuring the gases above the soil, so that the atmospheric gases, and then the gases within the soil, and you're looking at the difference in how quickly they can change. And that will tell you how much various mulches will interfere with or enhance the ability of gases to move back and forth. So we did bare soil, as I mentioned, that's the control. And I will preface this by saying that a bare soil in a bucket in lab is not like a bare soil outside. So no bare soil outside would be ideal. But we have to have something to compare all these mulches to. And so the only thing to compare them to is the presence or absence of a mulch material. And so bare soil is our control, even though this type of nice, light, fluffy bare soil would never occur in nature. Then we have a black plastic mulch, a single sheet. We used 4 inches of arborist wood chips, landscape fabric, and that was 1 single sheet of landscape fabric. Landscape fabric plus chips, which a lot of people do because landscape fabric's ugly and they want to hide it with something. A single piece of cardboard, and then cardboard again covered with chips, because this is a common way for people doing ecological restoration to do mulching, is they'll put down cardboard and then put chips over the top. And obviously there's many more materials we could have used, but when you're doing scientific experimentation where you're having replicates and having to have the space and time and ability to measure things, you get quickly limited to the number of treatments you can have. So this is how we collected the data. I just want to show you the equipment. It's not terribly exciting unless you happen to be really into electronics. But we had one machine that's called a Lycor gas analyzer, and that measures the CO2. And we also had an oxygen gas analyzer. And then the third picture, you can see it on mesocosm, now has a lid over the top that's keeping any kind of air that's outside the mesocosm from getting in and exchanging. So this is now a closed system just for the purposes of measuring. And so we can see that they're doing some measurements here. You can see them plugged in, and those are not for the gases. Those are some other things, and like the temperature and other stuff. And then the ports, you can see here, those little orange things are the ports. And so we took aliquots of air out of those ports using a syringe. So here's the one that's below the soil. Here's the one that's above the soil. And then those aliquots were put into either the carbon dioxide gas analyzer or the oxygen gas analyzer. So that's basically the setup. So this is, mostly a data generating experiment with, it looks like a whole lot of information that perhaps wouldn't be applicable to us in the landscape. But in fact, there really is. And I I took the data from our paper that we published and I made a chart out of it, which has really good practical application. So here's the chart. First thing I want you to see is that this is looking just specifically at a carbon dioxide diffusion coefficient. And a diffusion coefficient is a constant. So in other words, it's not a variable, it's not going to change. It's always the same. It's a measure of how easily carbon dioxide moves through each of these mulch materials. And so that's really valuable. So it's a good comparison you can make between mulches. Along the y-axis here, which is looking at the diffusion coefficients, this is an exponential axis. So each tick mark is a factor of 10. So think about pH scales or earthquake scales, and you know that when you go from a pH of 2 to 3, you're making it more alkaline by a factor of 10. So we have changes of factor of 10, and then we have our different treatments across the bottom here. So of course, the one with the highest diffusion coefficient, which makes sense, is the bare soil because it has nothing on top and it's nice and fluffy. It's not compacted. And so it's what an ideal soil would be, but it would never exist in nature. The next best treatment was the 4 inches of arborist wood chips. And if you look at where these bars max out, you can see that it reduced the carbon dioxide movement by not quite a factor of 10. But then we go to the one sheet of cardboard. It's 10 times worse than the wood chips. Landscape fabric is almost 100 times worse than the cardboard. And black plastic is over 10 times worse than landscape fabric. So when you step back and look at this, when you're making choices about mulching on soil, you can say that the plastic is 10, 100, 1,000, 10,000 times worse than 4 inches of wood chips. And there just was no question that the wood chip material, which is a nice, coarse, three-dimensional, chunky material, is much easier for gas to pass through than a sheet of something, whether it's cardboard or fabric or plastic. So this is the information you can take from this paper and then use that to explain to people, when you're making choices about, mulches to use, keep in mind the soil is a living system. And you have to be able to have gas transfer to have things stay alive, not just microbes. And this system was only measuring microbes. But when you have a complex system where you've got earthworms and plant roots and lots of other things that depend on oxygen, getting oxygen coming in and allowing carbon dioxide to get out, then when you're covering it up with the material that reduces that, you're going to have an unhealthy soil environment. So this is the paper that was published. You can get this online. It's it's old enough now that it's no longer behind a paywall, so it's easy to download. It's pretty much calculus based, and in that sense is really not very exciting to read. But what the abstract says and what the data in the paper support is that among the mulches tested, wood chips are a preferred method of mulching in terms of providing best gas permeability, particularly in landscape conditions. So this for me was the real benefit, because there hadn't been any testing of sheet mulches to look at the negative effects they have on soil. This is the first and only paper that's done this, and we can use this because it's extractable to use the information from it and apply it directly to a landscape, because landscapes are more complex than a microbe-filled bucket, and obviously there's more oxygen that's used in a complex soil landscape. Okay, so now we'll go on to misapplied science. So again, these are things that may have had at one point use in agriculture production or still do, but we're often encouraged through heavily marketed materials to use them in our gardens and landscapes. This includes things like anti-transpirants, Epsom salts, gypsum, hydrogels, and phosphate fertilizer, especially during transplant. And it includes practices such as amending the soil before planting, and by amending, I mean working material in and not laying it on top, and foliar fertilizer, so spraying leaves for fertilizer rather than applying things to the soil. So as I said, these are all things that at one level have some use in agriculture production, but have absolutely no use in landscape management. So I'm going to mention phosphate fertilizer because I think that in terms of chemical damage to landscapes, this is the #1 problem that happens to us as gardeners and happens to our clients, and is happening in cities all over the country, if not all over the world. And the reason is pretty simple, because we are encouraged to buy this because we're told that this will enhance the root growth of new transplants. And so anything that has a NPK where the P number, the middle number, is large, like this has NPK of with P being 15, is a phosphate fertilizer. And it also includes things like superphosphate, triple phosphate, and all of these things. You're told that it's great for feeding bulbs, it's good for enhancing root growth. And so people just kind of get used to throwing a handful of bone meal or whatever into the hole when they're planting, because they've been told they're supposed to do this. The problem is that if you don't know what you already have in your soil, it's really easy to start creating problems. And I will tell you that trying to get rid of excess nutrients is much more difficult than correcting a nutrient deficiency. So if you have a soil test done, whoops, sorry about that. If you have a soil test done and it says that everything is excessive and off scale, please don't add anymore. And what you will learn to do when you get these types of soil tests is you will be able to target exactly what, if anything, you need to add. And for 99% of the soil tests I see, maybe even higher than 99%, is the only thing that you might need to add is nitrogen. And that's nothing that they usually measure because it's a quite variable nutrient. But nitrogen is often added, especially during the summer when growth starts to slow down, because the nitrogen is becoming deficient. But most gardens and landscapes don't need anything else added. And so, but without a soil test, you just don't know. So what happens if you add phosphate without having a soil test to direct you to do so? Well, there's several things. The first is that phosphorus is going to compete with the uptake of both iron and manganese, and possibly other nutrients too. This is one of those areas of research that's fairly young and quickly growing in terms of looking at nutrient toxicity as opposed to nutrient deficiency, which people have studied for decades. And so what happens if you have reduced iron and manganese uptake is you get this interveinal chlorosis, and I'm sure everybody's seen this before. Oftentimes it's blamed on pH. Oftentimes it's blamed on a deficiency of iron and manganese, but unless you have a soil test done, you don't have any clue what's going on. So I can tell you for sure that when you have leaves like this and they're yellow between the veins and the veins are green, that if you did a tissue analysis, you would find that there was a deficiency in phosphorus, in iron or manganese, or possibly both. But then you have to say, okay, is there a deficiency in the soil? And most of the time when you do a test, you will find that there's not a deficiency of these elements in the soil. It's something else going on. And in this case, it's too much phosphorus. The second thing that happens is that when you use too much phosphate, you will not have mycorrhizal fungi colonizing your plant roots. And we know enough about mycorrhizae now to know that they are the rule rather than the exception. Nearly every woody plant has them, and many non-woody plants do as well. And they enhance uptake of water and nutrients for their plant hosts. If you don't have the mycorrhizal fungi, what happens is that the roots now have to work overtime. And what I mean by that is they have to grow more fine roots and root hairs to take up the water and nutrients that the mycorrhizae would normally take up. So this is kind of the nucleus to the myth. People observed that if you added phosphorus, that the roots seemed to grow better, but that's because they didn't have any mycorrhizae. So you're getting excess root growth because the mycorrhizae fungi aren't there, and that means the plant's spending more energy putting out new roots and less energy, with above ground growth, and that's not good for either the plant or for our enjoyment of the plants. The third thing that happens is that you will get aquatic pollution from too much phosphate being added. And all gardeners know that the nutrient that's usually most limiting in their gardens and landscapes is nitrogen. But in aquatic systems, plants are limited by phosphate. So when you get phosphate pollution in a waterway, whether it's a lake or a stream or creek or whatever, you will be rewarded with an algal bloom. And everyone's seen algal blooms before. You'll get this big, big cloud of green or sometimes blue-green algae that appear using up all that phosphate. And that seems fine because, of course, they're also releasing oxygen. But the problem is eventually phosphate levels go down. And when the phosphate levels go down, then the algae start to die off. When the algae start to die off, then the bacteria start decomposing the algae and they go up in numbers. And the other thing that bacteria do is they use up all the oxygen in the water, and then the fish die. So phosphate's not killing fish directly, but it starts a cascade of events that will eventually lead to fish death, and it happens every single time. So what we need to do is be looking at the research on phosphate use, and as well as getting soil tests and understanding what happens when you have toxic levels of certain nutrients. And so this particular paper here, again, it's something that's available on my web. If you look at my handout, there's links to where all my papers are housed online, and you can read this. You can see the work that's been done looking at the perception that, or not the perception, but the evidence that phosphate toxicity causes deficiency of iron and manganese in leaf tissues. And then we get to over-extrapolated science. And this actually is my favorite because, you know, it's being able to see that some things work really well in the lab, but you can't take them outdoors and expect them to work too. And this includes things like corn gluten meal as an herbicide. It includes a chemical called harpin, which was thought to be able to turn on plant defenses so that they would become immune to pest and disease attacks. And it worked great in the lab, did not work outside. And probiotics, so things like mycorrhizal inoculants and beneficial bacteria, they're heavily promoted because, yeah, they work great in a lab where you're working with a soilless medium that has nothing in it. But when you're working with a landscape or garden soil that already has a pretty healthy bacterial and fungal population, these inoculants are superfluous. They're not needed because there's already a good, healthy population of beneficial microbes in the soil. And then there's phenomena. And this includes the concern about allelopathy, and that's actually the one we're going to talk about today, as well as humus formation. So these are things that people are convinced are important in terms of how soils or landscapes function, but the evidence for them is really limited to lab situations. And so we'll look at allelopathy to find out why. So this is, I'll tell you, this has been one of my favorite stories in terms of debunking a myth. And the myth is that juglone, which is in black walnut, will kill plants located beneath them. And this is because people have gone and have seen, you know, black walnuts that have these huge patches of bare soil underneath. And because of work that happened last century, they feel like they're fairly justified because there's been a lot of publications on this. So this started back in the, I think, 1930s. And it was when, you know, technology came along far enough where it could isolate and identify chemical compounds. And one of the first things that was isolated, identified, was juglone in black walnut. And people had thought for a long time, this goes back to pre-Roman times, that black walnuts would kill other plants. So this is not a new belief. And so the scientists were interested in this because of this idea that black walnuts, you know, had this toxic substance. And one of the scientists noted during this work that his tomato plants at home, as they grew closer and closer to his black, became stunned and or died. And so he said, you know, to himself, well, you know, here's the evidence. It's right here. This is happening. The black walnut is killing the tomato plants. They're closest to it. So this became a huge thing. Scientists are really excited about. They published lots of papers on juglone as well as other chemicals from other plants that were identified. There's an entire journal called Allelopathy Journal that's dedicated to nothing but talking about all the allelopathic chemicals that various plants can make. And obviously the benefit of being able to identify these types of chemicals is that you could use them in weed control. You know, it's a natural way of controlling weeds. And so that's been kind of the motive behind doing this. But the question is, you know, has this ever been shown to actually happen? So what allelopathy is, if you're not familiar with it, it's just simply the ability of plant extracts to kill other plants. And the way you test it is really simple. You get a bunch of petri dishes and you put an auger in there, you know, so that's the medium that the lettuce seeds are going to grow on. And then the auger contains different levels of the chemical you're interested in this case, juglone. And if you get this type of browning, where things are leaking out of the germinating seeds, that's allelopathy. So this one up in the corner here, that's the control, and it's clear there's nothing leaking out, but these other dishes you can see there's leakage, and so that would be evidence of allelopathy. The problem is that lab test, which is germinating seeds in agar, was then extrapolated to what happens in the real world with walnut trees and plants that grow under them, with no experimentation to demonstrate that it actually happens. At least that's what it appeared to me at first. So this is one of those things, we're human and we tend to jump to conclusions a lot, and that includes scientists. And so scientists had jumped to this conclusion pretty quickly. But we have to remember that just because two things are happening at the same time or in the same place does not mean that one is causing the other. So this is called equating correlation with causation, and it's a common error in logic. So we're told that this juglone comes out of the trees by various ways, and it's going to kill everything that it touches. But the problem is, as many people have pointed out, and as probably many of you have seen, there are plants that grow under black walnuts and grow quite happily. So how do you reconcile these two things? How can you say that allelopathy in black walnuts means that the juglone is killing everything that grows underneath it, but yet there's landscapes that have things growing under black walnuts? So scientists had a tough time with this, and so they came up with lists, and they came up with lists of plants that were tolerant to juglone and lists of plants that were sensitive to juglone. And I'm a plant physiologist, so I looked at these lists and said to myself, I don't understand what makes these lists different from each other, other than supposedly one list is things will tolerate juglone and the other one isn't, because they include everything. It's all different kinds of trees and shrubs and vegetables and flowers, and there isn't any physiological reason that one group was different from another group, because there just wasn't. So this is where my skepticism got turned on, because I couldn't come up with a hypothesis of how there would be this difference between sensitivity and tolerance. So the easiest thing for me to do then would be to go back into the literature to find, where these lists came from, because I assumed that they all came from research where people had been testing this and shown that certain plants would die exposed to juglone and other plants wouldn't. And it's interesting because, as I said, you can demonstrate that allelopathy happens in lab experiments. There are no landscape experiments that demonstrate this happens. In fact, there's been a number of studies that have tried to find juglone in the soil under black walnut trees, and they can't find anything. And probably the reason this happens, and this is what these researchers have speculated, is that because you're looking at a living soil that's full of microbes, that juglone, which is an organic compound, gets into the soil from fallen leaves and roots and hulls, and is broken down pretty quickly because it's a source of energy for microbes. And so the fact is you can't find juglone in the soil. So during this, I found this information, and then I was really interested in finding out where these lists had come from, because the lists have been reprinted in almost every single university extension publication on allelopathy and black walnuts that's out there. And I was able to trace it back to a publication from Cornell that was called Walnut Wilt, and it was published, I believe, in the 1950s. And I don't remember the name of the second author, but the first author's last name was Christ, C-R-I-S-T. And fortunately for me, I have a previous graduate student who's on faculty now at Cornell. And so I emailed her and I said, I'm trying to find this publication. I can't find it online anywhere. And I'm hoping that you have that you can find out from the extension folks, there at Cornell, if they have a copy of it. So about 2 weeks later, I heard back, and I got this email directly from, they have a librarian who does nothing but catalog their own extension publications. And she said, this publication doesn't exist. There's no such paper. There is no such faculty member named Christ, here at Cornell. We don't know, we really don't know what this is. So you have to understand that the sheer depth of this misinformation that has been printed and reprinted in both extension sheets and in scientific articles for decades is based on absolutely no experimentation. It's all been conjecture, people looking and saying, well, got some tulips under my black walnut tree. They're not doing very well. Therefore, tulips are sensitive to juglone. I mean, that's what it boils down to. And it's not just sloppy science. It's not science. And the reason that this was such a fun one for me, because this wasn't just, your average gardener doing this. This is scientists that should know better, that we're making these leaps of logic. So we don't have any evidence that this happens. And it's not to say that plants might not suffer under black walnuts, but it's probably because things aren't being watered enough. And that's because an established black walnut tree is going to have a massive root system that's going to be sucking up most of the water and a lot of the nutrients. And if you put other plants under there and you don't provide more water and nutrients, then they're going to die. So the gardens that you've seen that have black walnuts and they have other plants under them are well managed and they get plenty of water and they do just fine. But it's not allelopathy. And I've looked at other chemicals now in terms of things that are allelopathic, and I can't find anything so far where they have demonstrated conclusively that there actually allelopathy that occurs in nature. It only happens in petri dishes. So keep in mind the correlation between two variables does not mean that one is causing the other one. Whoops. And so there's a really great website that's called Spurious Correlations. And it has great visuals like this where it shows an almost perfect correlation between the per capita consumption of cheese in the US and the number of people who died by becoming entangled in their bed sheets. And so if you were going to raise the correlation to causation, then you would be cautioning your clientele not to eat cheese in bed. So the point of this website, which I think is so valuable, is that there's tens of thousands of examples there. They make people laugh. They kind of get people to step back and think about things logically. And even though juglone and allelopathy isn't necessarily a hot button topic, there are other things that have been erroneously correlated as being causative, that this exercise in learning whether or not something is actually a causative agent or not is really useful. So I like this website a lot. I encourage you to use it. And I do have a peer-reviewed publication on this whole thing too that goes into the story of me trying to find out the information on juglone. And as I said, it was pretty fun to do. It's a free download. And that brings us to the good and not so good science. This can include things that are good quality research, but poor reporting. In other words, the experimental design is sound, the data are collected properly, the results of discussion are presenting things accurately, or maybe not, or there's a problem in the abstract. So this is due to researcher bias most of the time because, as I mentioned before, scientists are human too. And one of the things that we like to do is hope and pray that whatever we've hypothesized is proven to be correct or has been supported to be correct. And so when we're looking at our data, we're kind of wired to look for things that support what we think is happening so we can show how smart we are. And so this means that, that we will selectively highlight certain things in the abstract as being really exciting results. And then things that aren't necessarily so supportive, we don't mention the abstract at all. It's not that they aren't there in the paper, but this is the cautionary tale that you don't just read an abstract of a paper and assume you've got the full story, because sometimes the abstract is not going to match the actual paper, and peer review messed up. And that's because either there is no peer review, and unfortunately there's more and more journals that fall into that category, and I use the term journal really loosely there. And sometimes it's because human failings, and I'll discuss how one of those worked. So sometimes there's papers that you can't get to. You can see the abstract, but the rest of the paper is behind a paywall. Those of you that are master gardeners or university employees, the nice thing is that you can use your university databases through the library, and you can get these things because most of them are accessible through your library, through the university library. So you can see the whole thing, and you can find out whether or not it's actually the abstract is actually supported with the data in the paper. So here's the first example, and I'm using gardening examples, obviously, and this one was near and dear to my heart because I've spent most of my research life researching mulches. And so the claim in this paper was that newly transplanted trees should not be mulched. which was a shocker to those of us that do this kind of work. And it came out in 2012 from the University of Florida. And the researcher there, Dr. Ed Gilman, who's retired now, is legitimately regarded as kind of the god of arboricultural research. He had an incredibly productive lab and produced all kinds of really good research and is highly regarded. So when this paper came out with this claim, with his name as the first author, it really shocks the rest of us that are also doing mulch research. So here's the methods for this experiment. So it's really simple. It's something that most of us could just do at home, really. You have containers that are filled with soil and either left uncovered or covered with mulch. And then they were watered to saturation, and water was measured, water loss through evaporation was measured for 3 days after watering. And so then you can see it's a very simple comparison. Bare soil, mulch soil, which one loses the most water? So the results is that the unmulched soil, so the bare soil, had a total evaporation on average of 3 liters per pot. The mulched soil had a total evaporation of 2 liters per pot. And if you were to make a graph of this, and I always like graphs because they just show things in more than one way, is that you could say that mulching reduces evaporation from the soil by 33%, which is great and is completely accurate. And that type of reduction in evaporation has been shown for decades in mulch research. So there's nothing wrong with the experimental design or the results. The problem is with the abstract. And the abstract says, given minor reduction in evaporation, parenthetically 33% is not minor to anyone who does any kind of irrigation, and reported disadvantages of mulch application close to the trunk, landscape managers might consider changing mulch application practices for newly planted trees. So, this is the perfect thing to use. If you decide that you're going to try to do some educational outreach where you're trying to get people to think about, critically assess an abstract after having looked at the paper. The first thing we'd do is say, okay, well, let's look at this experiment. It was pots of soil. There's no trees in this experiment. This isn't a landscape. These are pots of soil. And so you can't really be talking about mulching trees and newly planted trees in the same breath as you're talking about a simple experiment that had nothing to do with trees at all. But that's the problem. And this is one of the things that if you're doing peer review, most journals will have a checkbox for the peer reviewers that says, does the abstract accurately reflect the content of the paper? And this is why, is because the abstract is supposed to be the summary, and it's supposed to be an objective summary. Well, this has nothing to do with the research at all. It's somebody, with their opinion that has nothing to do with the particular research. So one of my peers, who also does mulch research, was pretty highly incensed with this whole thing when it came out. And this was his statement. The Florida study was conducted in a highly artificial system, was very narrowly focused, and reflected water loss over a period of 3 days. We have conducted field trials with real trees and shrubs growing in real soil where we have found real benefits in terms of plant survival, plant growth, soil moisture, and plant water relations over the course of years. So he was talking all caps, and he's a pretty low-key guy, so when he goes all caps, he's really not very happy. So what happened? Okay, we have this researcher who had a career of excellent work in arboriculture and was published in a highly regarded journal having to do with arboriculture and urban forestry. So all I can tell you is what I think happened. Excuse me. When papers go out for peer review, they're anonymized, and that means that there's no listing of the authors, and the names of the reviewers are also kept anonymous, so the authors don't know who the reviewers are, and that's what's called double blind. So nobody knows anything about each other, ideally. The problem is that when it comes to work with mulches and urban trees and arboriculture, there aren't very many of us. And so when the peer reviewers were reading this paper, which had no names on it, but when they got to the methods section, where they talked about this work being done in Florida, everybody knew it was Ed Gilman's lab. And because Ed has a reputation for really good work, they kind of glossed over the abstract and didn't really think about what the abstract was saying and the fact that the abstract was biased. And what should have happened is that someone should have caught it and said, hey, you got to take this part out. It's conjecture. You can put it into your discussion if you want to, but it does not belong in the abstract because it does not reflect what happened. So that's the problem. So the warning here is don't succumb to SSS or single study syndrome. And what I mean by that is if there is a paper that comes out that's at odds with the majority of other papers, it must withstand increased scrutiny. And that's because science changes all the time. No one's denying that. And that's, again, why my field is so exciting, because there's always new stuff. But if something is going to change the dialogue about a particular subject in the field, it has to meet a really high bar and has to be repeated by other people before it becomes the new opinion in terms of, in this case, mulches. And it doesn't, because there's just a huge, it's a robust and decades old body of literature that shows that mulches help retain water in the soil, and that they, if correctly applied mulches, are not going to hurt newly transplanted trees. So that's why you have to be really careful about reading just the abstract. Okay, here's another one. The claim is that only native trees can provide resources for native insects. And this came out in 2009, and again by somebody who is well regarded in the field, Doug Tallamy, who is an entomologist. And so he was looking at Lepidoptera larvae and how they used native versus introduced plants. Okay, so keep in mind what the comparison is, native plants versus introduced plants. So the methods in this paper is that they labeled all genera, okay, so these are all the genuses, comprised of native species with geographic distributions in the mid-Atlantic region as native, even if the genus contained one or more species that had been imported for use from Europe or Asia. Well, now they've already screwed up their own study, because they're no longer comparing native and introduced. They're putting a bunch of introduced plants into a genus they're calling a native genus. And that's to me, this is a fatal flaw. But it was published in Conservation Biology. There weren't any plant scientists that were in this group. They're all entomologists, and this didn't get caught. So let me show you what happened in terms of this error in experimental design. So this meant that there's almost 200 introduced woody species, so that means non-native trees and shrubs, that were included as native in the genera that were reported as native. And then the second problem they had was that this analysis was extrapolated to all herbivorous insects. So the lepidopterans, which are, as you probably know, contain a lot of specialist species that have, like monarch butterflies, for instance, that have to have milkweed. Lepidopteras, many of them are specialists. But this was extrapolated to all herbivorous insects, specialists or otherwise, with the statement, we can think of no reason why Lepidoptera would not serve as reasonable surrogates for other folivorous insect herbivores, which is stunning to those of us that worry about things like elm leaf beetle and all these other introduced insects that can eat just about anything. So there's a good reason why this shouldn't be extrapolated to all insects, because not all insects are specialists. So here's their chart of the 20 most valuable plant genera in terms of being able to support Lepidopteran species. And they include things like Prunus and Rubus and Rosa. And I don't know about the invasive species that you all have in North Carolina, but I can tell you that all three of those genera here include highly invasive species, not just introduced, but invasive species that are heavily used by insects. Here's a good example. This is Himalayan blackberry, and it supports a lot of our rare insects. This is a white-bottomed bumblebee that you never see very much in Washington State anymore. And the first time it was photographed for decades, it was on, a flower of Himalayan blackberry. So the point of this is twofold. One is that you can't fold all these non-native plants into a native genera and then say they're all great. The other thing is to realize that insects adapt. Maybe not all insects, and certainly not all specialist species, but many insects adapt to new food sources, and that's why things become invasive. not just because they grow well, but because things spread their seeds or they, spread pollen and enhance fertilization. So the whole thing was really poorly designed and flawed in terms of its interpretation, because it did this combination of just folding all of these non-native species into a genera that they had labeled as native. And I actually talked to a guy who had been a graduate student of Doug Tallamy's and was familiar with this study. And he told me that one of the reasons, at least, that they had folded all these trees in there is that they didn't have any plant people on their team and they couldn't distinguish between the species well. So there's another reason why you really need you need experts with all of your various things. If you're working with insects and plants, you better have some plant scientists as well as entomologists. Whoops, sorry about that. Okay, so those are two examples, and those are of people that are highly regarded, that do good work, but sometimes because of their biases, they screw up. There's some that are much worse, and this falls under the poor quality research tag. It's common with authors who have absolutely no business publishing on what they're publishing on because they're not experts. So when you're doing the crap analysis, this is the credibility part. Is this a credible source of information? Oftentimes they conflate correlation to causation, and oftentimes it's published in very marginal journals, often with no peer review, sometimes what we call predatory or pay-for-play journals. And so they're not good sources of information. Fortunately for us, it's pretty easy to find out the quality of journals online just by doing a little Googling and finding out whether or not these are peer-reviewed and whether they're highly regarded by experts in the field. So here's an example. Neonicotinoids cause colony collapse disorder. And so this was back in 2014, and we were having a lot of issues with collapse of European honeybees. The irony that people were concerned about honeybee colony collapse when it was an introduced species never escaped me. But this was obviously something that was of concern to farmers that were using honeybees for pollinating their crops. So because of the concern with neonicotinoids, and there are some legitimate concerns with that, this paper came out and was of immediate interest because of the connection with collapse disorder and neonicotinoids. So we'll do the crap analysis on this shortly anyway. So we look at the credibility of the authors. The first author is at Harvard, so that's just fine. The other authors are beekeepers. This is not a bad thing. Oftentimes when we're working with landscapes or field situations, we have to be using someone else's land, and we would not be able to do the work without them. And that's the definition of when someone should become an author. So if you can't publish the work without someone's assistance in some way, shape, or form, then those people become authors. So that's not a bad thing. What is a bad thing, though, is that first author has a PhD in public health. Okay, public health is not entomology. It has nothing to do with understanding anything about pesticide application. There's absolutely no relationship between a public health degree and anything covered in this article. So that's a huge red flag. It's somebody who, just because they have a PhD, has a self-inflated sense of self-worth and decides that they can publish on anything they want to. The article is written in very poor English. This is a red flag. I'm actually an editor of a magazine and 2 journals, and so this is something I'm a real stickler for, is to making sure that the English is as impeccable as I can make it before it goes out to print. It may sound, kind of entitled, but that's just the language of science. So currently the language of science is English, and people that want to have their work widely read by scientists all over the world will publish in English journals. So if it's not in good English, that's a bad sign. The bees themselves were forced to feed on high fructose corn syrup with high levels of neonicotinoids. And what I mean by forced to feed is this was the only thing they had available to them. Normally, when you're doing experiments with insects, you do what's called choice experiments. And so they would have a choice of what to feed on. Well, these insects had no choice. They either had to eat this stuff or they would die. And as several entomologists pointed out online, the levels of neonicotinoids that were being used were far above what would be found in the real world. And then the capper to all of this is that there were no statistics whatsoever to show any significance to this study. It was just a really bad study. And so that's when, if you're doing your cap analysis, your BS meter, BS standing for bad science and among other things, goes off scale because it's just bad science. Okay, and this is going to lead to my last one that I'll discuss, which is glyphosate causing human disease. And it makes no difference, to me, whether or not you or a client or whoever wants to use glyphosate or not. That's a choice thing, and that's just fine. But the problem is that the hysteria around this chemical. And it all started with this paper published in 2013. I'm not going to read the title because it hurts my throat and it hurts my brain, because it's one of those things that is just indecipherable, especially if you're not a scientist. And if you want to really boggle your mind, just try reading the whole thing. And I can say this as a scientist, that I couldn't understand most of it because it had so much jargon and just nonsense in it. And it's done deliberately. It's done to bamboozle the reader into thinking, boy, this person's really smart and I don't understand what they're saying, but they're a scientist, and so I'll believe what they say. So let's look at this one in detail. Well, first of all, we know that because of the work of this particular researcher and her various cohorts has led to all kinds of hysteria about what glyphosate does, including causing obesity, Alzheimer's, autism, anorexia, dementia, inflammatory bowel disease, depression, Parkinson's, reproductive issues, liver diseases, and cancer. And people have bought into this. Juries have bought into this. Juries are not decided by science. They're decided by whoever has the best lawyers and the most credible witnesses. So the unfortunate thing is that, you have these lawsuits that are decided based on emotional appeal. They're not based on science. And that's just led to some very unfortunate things in terms of the perception of what glyphosate is and what it does. So let's look at this article in detail. The author of interest is Stephanie Seneff, and she's at MIT. She has a PhD in computer science. So she doesn't even have the credentials of working with living systems like at least the guy in public health did. She does computers. She doesn't know anything about any branch of biological sciences or chemical sciences for that matter. She has no credibility as a researcher in this field. It was published in the Journal of Entropy, another one of those twists of irony that I just love, given what entropy means. It is a non-reviewed predatory journal. You pay enough money and they will publish it. And this is where they publish their stuff, is in this kind of these open access garbage journals. Not all open access journals are garbage, but this particular one is. One of the quotes in there was that glyphosate is a textbook example of exogenous semiotic entropy, the disruption of homeostasis by environmental toxins. Now you can see what I mean about trying to read this article, because it's just this gobbledygook that just it makes your head feel like it's full of cotton. But since she said this was a textbook example, this phrase, exogenous semiotic entropy, I took that phrase with quotation marks in 2013 and stuck it in Google search engine to see how many examples of exogenous semiotic entropy I could find, and I got one hit, and it was this article. They made it up. They made up this textbook example thing, and this is so egregious. Sometimes I just find myself completely speechless. And then it gets even worse because, of course, this speaks very deeply to a certain segment of people who don't like what they perceive as big ag or big pharma or big whatever. They don't trust chemistry, and they think that that there's just all these really bad chemicals out there that are killing everybody. And so this one particular group got a hold of Dr. Seneff and basically was asking her to interview with her to say, how did you do this remarkable work? All these scientists have been researching it for decades and they didn't figure out what you did. And this was her quote. So basically what I do is I read papers and I process them with the computer to help me understand them and interpret them and generalize and build a story. So she just makes stuff up. It's all she does. It's garbage in, garbage out. And it's it's inconceivable to me that anybody who considers himself to be a scientist at a university would do this kind of stuff. I don't understand what drives her to do this. It's just, as I said, I'm speechless. But this is what happens, and she continues to publish this stuff. And it continues to be used in courts of law for determining all these damages from people who say that their injuries or their health issues are due to the fact that glyphosate is used more now than it was in the past. Because you have charts like this, where it shows that, hey, glyphosate use has gone up, and gee, autism has gone up too, so therefore the glyphosate is causing autism. So this is taking the conflating correlation to causation to incredible heights. And that's why that website about spurious correlations is so important, because there's absolutely no science, research, anything out there to suggest there's any link between these two things, but this is what she does. And the sad thing is that it's really freaked out a whole lot of people about glyphosate, which is the active ingredient Roundup and other types of weed killers. And the thing is that if you know even the slightest bit about glyphosate and how it works, it interferes with a plant biochemical pathway for making things like lignin and tannin. It's a pathway we don't have. It doesn't affect us. But all of the facts about it are lost when you have this kind of really bad science with quotations publications coming out. So anyway, how can you find good information? I hope I've given you some ideas about that. As Debbie mentioned earlier, I do have my website that has things like my horticultural myths, which are white papers, and other things like podcasts and other fun stuff, all science-based. I mentioned the scientific literacy guide. I do encourage you to download that and use it as you like. I've published a number of books on horticultural myths, as well as sustainable landscapes and gardens, how plants work, which is kind of plant physiology for gardeners, as well as a series through the great courses on the science of gardening. And so all these things, some of them actually go through formal peer review, but even if they're things that didn't require formal peer review, I always send them out to my colleagues before I end up publishing stuff, because I don't want to be embarrassed by having something that's wrong. My ResearchGate library link is on the handout, and what ResearchGate is kind of a website that mostly researchers use to kind of communicate with each other and share their work. But anybody can create an account. It doesn't cost anything. And once you have an account, then you can search for any researcher and find out what papers they have. And we just kind of use it as kind of a online clearinghouse for just collecting all of our papers so people can find them and download them. So almost all of my papers are on there. The Journal of NACA is something that is published through the National Association of County Agricultural Agents, and I've been publishing things there with a colleague on garden myth busting. So this is a peer-reviewed journal as well, and so we've done things on landscape trees, soil structure and functionality, soil nutrition, pruning woody plants, and then the last one that we did last December was on arborist wood chip mulches. And so that's a good source of peer-reviewed information that is much of the information I've been telling you, but with all of the citations to the literature to support the various statements. This is our blog. We've been doing since 2009, and every week there's a different publication up or a different post up. We've got some great people on there. The agricultural meteorologist for Georgia. I don't know if you know Pam Knox or not, but she's one of our bloggers. There's me. We've got an entomologist from Montana. We've got a pathologist from California, vegetable specialist from Nebraska. We've got people from all over that will post on whatever they feel like posting. So that's kind of a fun place. And because it has a search box, you can go through and you can search on any topic over the last 14 years now to see what we've published on that. We've got a Facebook page, which is kind of a clearinghouse just for interesting information and sometimes fun stuff that one of my volunteers helps manage. And so she curates this as being a good science-based site on Facebook. And probably my favorite thing that I do on social media is manage, along with a bunch of volunteers, the Garden Professors blog group, which right now has almost 27,000 members, and they're from all over the world. And it's a science-based discussion group. People can post questions and oftentimes with pictures, to try to figure out, problems. We cover just about everything, and you'll have experts weighing in from all over the world. We don't allow things like do-it-yourself recipes or anecdotal experiences. This is just, the good science behind different issues that have to do with gardens and landscapes. So anyway, I'm out there along with a lot of other people, and we all enjoy working with people from all over the world. So with that, I'm happy to answer questions, and I hope you enjoyed it.
So I do have a bunch of questions. So I'm going to go way back into the archives here and talk about lasagna gardening again. So the first question was, this person says that she's used the lasagna method for initial bed creation and used newspaper instead of cardboard, and it really worked. It worked well for her. Is that an improvement over the cardboard? I know it wasn't part of your study, but.
Well, sure. I mean, anything that's not cardboard is just a really bad material because it doesn't break down easily. But what I would suggest is kind of imagining taking a single sheet of wet newspaper and putting it over your face and then trying to breathe through it. And that's what that plus more newspaper plus compost is doing to the soil, is reducing the ability for the soil to breathe, literally. So it might be better than cardboard, but it certainly isn't better than mulch. I mean, a good coarse 3-dimensional mulch. That's going to be better for gas exchange than any kind of sheet mulch.
Okay. And so the the very next question was about gas permeability, which is what your study was about. And obviously that's not the only thing that you decide that is a deciding factor when you're building a new bed. So what is a good approach when you're also trying to hold moisture in, trying to get the gas exchange right, but keep weeds down? Is there a study that talks about what's kind of the happy medium between all of those things and, well, anything else you'd be managing?
Well, the best thing for keeping weeds down and for beds is a thick layer of arborist wood chips. I've published work on that. Other people have too. Keeps the weeds down, keeps the moisture in, promotes root health. And there are many published papers on that, not, as I said, not from just me, but from other people too. A lot of people don't like the way it looks, but I will tell you that the time you spend weeding will go down to just about nothing, and that you'll end up spending more time pruning because things are growing so much faster and better than they did before. And I actually use wood chips in my vegetable garden as well. I use them as kind of overwintering rather than planting a cover crop, letting the soil kind of rest for the season, and also keeps the weeds out. So really, for keeping weeds out, there is nothing better than a thick layer of a coarse woody mulch like arborist wood chips.
So when you say thick, how thick?
You have to maintain it about a minimum of 4 inches. And the
Okay.
reason is because it keeps sunlight away from the seeds so they can't germinate. So once it gets below 4 inches and the sun can kind of get through there, and you'll start getting weed growth again.
So this next question was asked, a while back. So you have given us a lot of fodder, if you will, for this question. How can we combat some of these myths? And this person mentioned like the whole native plant thing that has almost a cult-like following.
Well, so go check out things I have online. In
Right.
terms of the native plant debate, I actually published an article in Horticultural Urban Forestry back in 2015 that kind of was a scholarly critique of the literature on this. I had about 140 papers I looked at comparing the effects of native and non-native woody plants, so trees and shrubs, on landscape biodiversity. And the result of that comparison is that plant nativity plays nearly no role in how biodiverse a landscape is. What's important is diversity of plant species, vertical structure, accessibility of water. I mean, there's about 9 different factors that were important. And so that paper, along with the fact sheet that I did based on that paper, is really useful for looking at it. And as I said, it's all based on science, and it weren't my studies. I was just kind of analyzing all these other people's studies and drawing conclusions from them. So I mean, people have to be receptive to it, though. I mean, if you've got people that have bought into, a belief, and they're not going to challenge that belief, it doesn't matter what kind of science you have, they're not going to listen to it.
Yes, that is true. So this next one is about a completely different subject, watering. This person said that, and we've all heard this, that watering when it's sunny can cause leaf burn. Watering when it's hot, you mentioned that, you're never supposed to do that. So of course around here, I'm not sure what that means. We're never supposed to water, I guess. So what's the truth? Because it just rained and it was really hot today. So I'm pretty sure it was hot when it rained.
Yeah, well, I have on the blog, when you get to the blog and do some searching there, you can find a paper that I dissected on that particular topic. The fact is that it rains all the time when it's sunny out. It does not scorch the leaves. There's this perception that a leaf droplet is going to act like a magnifying glass and scorch the leaves. It does not do that. What happens is that if it's been hot out and people see their plants wilting, and then they go out and water things, oftentimes those plants have already experienced terminal wilt, and the next day they're going to be brown and dried up. But that's not from the water, it's the watering, it's because they were left to dry too long. So what I tell people to do when they're worried about watering is to have one plant that's kind of their canary in the coal mine. So for me, it's my hydrangeas, because they have great big flat leaves, and as soon as those things, the younger tissues start to wilt, that's when the water goes on. You let them go too long, then you're going to get terminal wilt and you will get the browning. And it's that whole thing where you're correlating watering and brown tissue. Oh, it must have been the watering that caused the brown tissue.
Good point. And hydrangeas are an excellent choice for the canary plant.
Yep.
Absolutely. So any other questions, everyone? I have posted a link to the handout, and that'll stay there for a while, actually. So
Oh, great. Yeah, and I was just going to mention that if people want to join the Garden Professors group, which we really love having new people join, you will have to answer a few questions, and that's just because we are trying to keep, bot accounts out. And so you have to make sure you answer the questions to be led into the group. And that's the only thing that would keep you from getting in.
Oh, yeah. And a lot of what I consider the better Facebook groups are kind of like that. We like that. Yeah, so Rich asked if this is going to be recorded and can he share it? And yes, it is. And that link that I gave you for the handout, right above that is in the same link, right under the September 7th header. That's where the presentation will be. And like I said, I'll try to get that as soon as I can. And I put it on our YouTube channel, so then you can see all the other things that we've been.
Oh, that's great. If you wouldn't mind, send me a copy too, and I'll put it up on our Facebook group.
Wonderful.
If that's okay.
Absolutely. Yeah, we love that. Yeah,
great.
That'd be great. Okay. Well, you had several comments about how helpful and entertaining and good this was. So
good.
I think you've got some new converts there.
Well, all I can say is that I really enjoy getting people excited about, doing the detective work behind information to find if it's good information or not. It's really very rewarding to learn how to do that.
Absolutely. Absolutely. And sometimes you find that the lazy gardener approach might actually be the best.
Yeah.
What I'm learning. Good thing that's what kind of gardener I am. Okay. Anything else, guys? Nope. Okay. Then I'll post this as soon as we can. And thank you all for hanging out. And she says the link doesn't work. Okay. Let me check that. That's because you're going from if you'll just copy that link and put it in your browser, it should work fine. Sorry about that.
Oh, and I was going to mention, if you don't mind, so there's a program in South Carolina, and I think it's in February, called The Joy of Gardening. And I'll actually be speaking at that next February. So if there's people that I can't remember the name of the town, but it's actually close to the border of North Carolina. So
is it you said in February?
I think so.
I think it's the big expo that they have down there. I went to it last year. A couple of us have been down there before, so we'll be looking for it. That's great.
Yeah, it's the joy of. Yeah, it's called the Joy of Gardening. It's in Richburg.
Richburg, yeah. That's exactly it. Yeah,
we'll be there.
Kelly Norris was there last year, and a couple other people. So it's a great thing to go do because there's nothing else to do in February gardening wise. So you might as well go learn some new stuff. That's
The
awesome.
link to your site at Union County does work, but the link within your site that says to get the handout does not go anywhere.
Okay, then I will fix that right now as soon as we get off
Thank you.
here. Okay, thanks. Okay, thank you guys. Good night.
Thanks very much. Nice to visit with you.
Yeah, thank you. Bye, everyone.